Okay, so with the immune system, there were two parts to it, innate and acquired immunity. So innate immunity was what we did on Thursday, which is just generic, more generic ways to fight against a pathogen, not specific to one pathogen versus another. And then acquired immunity, which is very specific immunity against a specific antigen on the surface of your pathogen. And so acquired immunity, that specific immunity, involves two major types of cells, B cells and T cells. B cells is what we talked about on Friday. So remember, let's just quick little review. When you come into contact with an antigen for the very first time, your body has to select which B cells, so you have this army of B cells, as to select which B cell will work against that particular pathogen. How does it know? The B cell that's selected has to have a complementary shape on the ends of the receptors that will fit with that particular antigen. So out of your thousands of different B cells, of which you only have a few of each type, you figure out which one is going to work against that antigen, and then that triggers the B cells to make clones of themselves, so it divides, and then differentiates or specializes. Some of those become memory cells, some of those become plasma cells that secrete antibodies that will fight against that particular antigen and only that antigen, not another one. The purpose of the memory cells, what's the purpose of the memory cells? Maria?
They live throughout the rest of the life and are there in case that antigen returns and they're ready to attack that antigen.
That's right. Yeah, so the second time you come into contact with the same antigen, you've already got these memory cells, so you don't have to go through this process of the selection and the dividing and the differentiation and so on. You don't have to go through that process again, which takes time. And so therefore you can launch your attack against that pathogen much quicker the second time around. And so, did we talk about this graph?
No.
No. Did we write this?
No.
So in the secondary immune response, that's the second time that you come into contact with the pathogen, your memory cells facilitate a faster, more efficient response. And so we look at this graph here. You're looking at time in days, and then along the y-axis is your antibody concentration. So how many antibodies are present? So let's look at my blue line here first. There's two different lines on here. Let's look at this one here. So I'm going to put an arrow here to illustrate about right here is this person's first exposure to antigen A. So that means in their lifetime, right here, what about day 2 here in this graph? It's the very first time they came into contact with that antigen ever. So then notice that several days go by and there's no antibodies present. But then, after those several days, you start to get in the body antibodies produced. And so, and the antibody production goes up and up and up. All right, so here's your first exposure. Can somebody, actually I'm going to have you talk with the person next to you and figure out a reason why you have this gap from when you first are exposed to the antigen to when you start to produce antibodies. Why is there a gap?
Because they have to find what B cell matches that antigen, and then it has to slow itself, and then it divides it. How does it take us to so many cells to find that one? Oh, we have to see the antigens. So that takes a while, and they have to go for I think there's a whole process called
Oh, yeah, it is. I tried to put this one. I don't remember really straight.
I like how I think I would be more opposed to her writing. I don't really
When it spikes up again, this would be like second year. Right at 28 days. Can't walk there.
Okay. So, we have this lag between the first exposure and the buildup of antibodies. Why is that, Devin?
So like know which antibody release.
Okay, so it needs to know which antibody to release, and what do you mean by matching?
Match the receptor.
Yeah, absolutely. So going back to this picture here, when you first get this antigen in your body first has to match up the antigen, that shape, or the epitope, if there's more than one shape on the antigen here. Epitope is one of the shapes there, with the correct B cell, and then stimulate that B cell to divide and then differentiate into your plasma cells, which then have to secrete antibodies. So that whole process from when this first gets into your body till eventually you get this clone of plasma cells to produce antibodies takes time. And so therefore, because of that, you don't see the antibodies starting to go up in concentration until days after that antigen first gets into your body. So you have this lag here. So then the antibody increases and then eventually fights off the infection. So this is antibody to antigen A. And so it gets rid of all of the antigens in your body, and so therefore the antibody concentration goes down. They're done its job. And so right here, this curve right here is what we call the primary immune response. So antibodies go down in concentration. All right, so then going and looking at this curve now, all of a sudden we get a spike. What would cause you to get a spike? So your antibodies go down because we got the antigen out of our body. Yeah. The antigen comes back. So right before this spike, right here, I'm going to put an arrow here. This is going to be your second exposure. To antigen A. So you've gotten exposed to it a second time. And so after you're exposed to it a second time, look at here, your antibodies spike up. So, and then look at there, you have way more antibodies the second time than you did the first time. So the response the second time is greater. You get way more antibodies, and it's also what in terms of time? Faster. You don't have that lag that you did here. Why don't we have that lag? Because as part of the primary immune response, you not only made plasma cells to make antibodies, but you also made memory cells. And these memory cells can produce these antibodies much faster. So this spike here is what we call our secondary immune response. This is faster than your primary and produces more antibodies. Sometimes they call it, it's an enhanced reaction, so meaning it. But really what that means is you have more antibodies there. And so oftentimes, if you come into contact with that same antigen, if you have your memory cells, it may, your immune system may act, respond so fast that you don't know even that you had the antigen in you. So you don't even get sick or whatever symptoms that particular antigen gives you. Shane? Yep.
Okay. So like kind of back to like the first exposure, when Katie and I were talking, she brought up the thing about trying to find the certain receptor. Couldn't it also be like there isn't enough bacteria for like your cells to recognize that it needs to do something, or like whatever the antigen is?
Yeah, like if you only have one single bacteria and it hasn't gotten into, let's say, so oftentimes it gets into your lymphatic system, which then, like, you have B and T cells in your lymph nodes, and that's a lot of where that selection occurs. So yeah, it may take some time to actually get to the spots where it will come into contact with your B and T cells, or in this case B cells. We're talking about B cells, but so yes, so there's that too as well.
Which would take longer, like for the certain amount of bacteria to be like grown, or to find those stuff.
Well, it's not, they're not isolated events, you know what I mean? So you get...
So it's like kind of a mixture of both having a...
Yeah, because you get this bacteria in you, and bacteria naturally divide. So, like, they can be dividing while moving throughout your body to get to where the B cells are to do the selection process and so on. At that point, there would just be more of the pathogen to, you know what I mean, launch the attack. So I guess both of those are occurring simultaneously, all right, to get to the B cells to select for them. Now the picture here has this red line here. What do you think the red line, why do they put the red line here? What are they trying to show you with the red line? Katie?
Could it be that there's a different strain of that bacteria, or it mutated, so you're getting the response to the first one, but then the other part that mutated is also affecting it?
Yeah, so if this is, when it says antibodies to B, this is a different antigen. So as Katie said, the different antigen could be because, let's say this is an antigen on the surface of a bacteria. This bacteria mutates, it could affect the antigens on the surface of it and therefore change them. And if you have a different antigen, then your immune system recognizes it and you'll have another primary immune response. So your secondary immune response and any immune response after that is only to this particular antigen. So this could be a different bacteria altogether, different species of bacteria. This could be a virus, but or it could be this bacteria mutated. Either way, the antigen B has a different shape than antigen A, and therefore you have a unique new immune response to that. So therefore your primary immune response is to one specific antigen. And so when you come into contact with different things, you have a different primary immune response for all those different antigens, and therefore make memory cells for all of those different antigens. So one exposure, that's one exposure to one cold virus is not going to make you immune to all cold viruses. There are lots of different cold viruses out there, and viruses mutate very quickly. They change very rapidly, so the antigens on the surface change very rapidly, which is why you can constantly every year get colds and multiple colds. It's not that your immune system's not working. You do have memory cells against the cold virus that maybe you had last year, but now you've come into a different virus, a different respiratory virus that is giving you same symptoms of the cold, but it's a different virus with different antigens and so on. And so you have a new primary immune response. So therefore you have that lag time, which gives you enough time to get sick, and then your body has to go through that primary immune response and heal from that. All right, good. All right, so I guess I should write here. So this is right here is your primary immune response to antigen B. So key there is it's a different antigen. And so where would my first exposure to antigen B be, do you think, on this graph? Roughly.
Probably like 28 or a little bit before where it starts to come up.
Okay, probably around 28, all right, somewhere before where it starts to go up here. So, I mean, it's not an exact science here, so I'll just put here first exposure. It can range to antigen B. Ready? All right, so that's your B cells part of your acquired immunity. So now we have to look at T cells and what's the job of the T cells. So first of all, I'm going to introduce a couple new words to you: humoral and cell-mediated immunity. So humoral immune response involves activation and clonal selection of the B cells. That's a review. That secretes antibodies. So, and then cell-mediated immune response involves activation and selection of cytotoxic T cells. So, what we've already talked about this, the clonal selection of B cells and secretion of antibodies. We just didn't put this name, humoral immune response. So when we talk about acquired immunity, just to give you a big picture here, it's divided into humoral, the humoral immune response, and the cell-mediated. And so we've already talked about humoral. This activates B cells. And cell-mediated activates cytotoxic T cells. We have to talk about what these do. And they're both involved in this immunity. The B cells attack against, these guys attack against the pathogen. So that's one way to get we want to get and get the pathogen out of the body. But the cytotoxic T cells, this part here, has a different role. These guys attack infected cells. So there's two launches of attack here against the pathogen itself and any cells that the pathogen has already infected. So we want to get rid of that. So this part here on the left is what we talked about on Friday, B cells and antibodies. So here we're going to look at the T cells and how they get rid of rid your body of the infected cells. So you have here a picture that shows you the actual cells and what happens here. And mine is divided in half here, so is yours, but mine's color-coded. On the left is that humoral response, and the B cells are colored here as blue. So this is what we just did. So your body gets infected by an antigen, and an intact antigen activates your B cells, which then triggers your B cells to divide and differentiate into plasma cells, which will secrete antibodies and help to fight your infection, and memory B cells. So that part we've already talked about here. So it's this part here with all of these T cells that we're going to talk about today. And there are two main type of T cells that are involved in this. Helper T cells and cytotoxic T cells. And so we're going to look at the role of those. I just said here that cytotoxic T cells are going to help with infected cells. They have a direct, notice here, down here, defends against infected cells. So these are the two ways that we rid our body of them. The cytotoxic T cells help with this part. Notice the helper T cells don't have a direct anything that they do to actually defend your body. What they do is they help or support other cells. So they help support B cells, they help support cytotoxic T cells so that they can do these two things. And so we're going to start with the helper T cells and look to see how they help. So, helper T cells are a response to nearly all antigens. It says the surface protein called CD4 binds the class II MHC molecules. This binding keeps the helper T cell joined to the antigen-presenting cell while activation occurs. Activated helper T cells secrete cytokines that stimulate other lymphocytes. So antigen-presenting cells, I'm going to abbreviate APC from now on. And lymphocytes, what are two kinds of lymphocytes? That are involved in this? These are your B and T cells. Alright, so you have a picture here. Underneath it I have a couple of, let me get them started. Underneath here I have some descriptions as far as, mine is color coded here, the steps 1, 2, and 3. So if you want to write those down, I'm going to give you a few minutes to do that, and then I'll talk about it. And so I'll dim the lights to make it easier to see. Okay. says helper T cells and the APC, which is the antigen presenting cell, are joined. So here's your APC, your dendritic cell, and it's taking in the bacteria. So I'll put here behind here APC, that's your dendritic cell. And it's taking in the bacteria and engulfing it. And remember that this is a good thing. This is part of your innate immunity where these cells take in anything foreign and destroy it. So that's a good thing that it's doing that. And then what it does, in addition to destroying the bacteria, is take some of those antigen fragments from the bacteria and present them. So this is right here, uses this MHC molecule presenting this antigen fragment. So this is the presentation presenting the antigen fragment. Right there. All right, so that's the MHC presenting that. All right, so that's what distinguishes this APC from others, is that this says, hey, I've just destroyed something foreign. So therefore, we need some more helpers, and besides just this innate immunity. And so in comes the helper T cell. The helper T cell comes over to the APC, and two things happen as a result of that. The APC stimulates the helper T cell by doing two different things. When the helper T cell binds to it, that's one way that this cell is communicating with the helper T cell. The second way it's communicating is the APC releases those chemicals called cytokines. These are chemical signals. And so these are signal molecules that this cell is releasing to communicate with this cell. So what must this cell have? Receptors, right? So this has receptors on it that these cytokines can bind to, and that triggers, remember from last chapter, a signal transduction pathway, which then triggers a response. What is the response here? This helper T cell will divide and make more helper T cells, and also will start to release cytokines. So they're releasing their own chemical here. And so that's what the first couple of things, first bullets here are saying. So the APC releases the cytokines, then that binding to the APC and the cytokines that were released from the APC stimulate the helper T cells. So then it's releasing cytokines. Notice here there's two directions of the arrows from the cytokines. The helper T cells actually stimulate themselves. So they release these chemicals which communicate with themselves. What that does is stimulate them to divide and make more helper T cells, which then can make more cytokines, and so on. Why do we want to make more cytokines? Because these guys help activate your two different types of cells, your cytotoxic T cells, which are going to kill your infected cell, and your B cells, which are going to secrete antibodies to get rid of your antigen. And so therefore, these two cells here must also have a receptor in the membrane that the cytokines can bind to. So these guys both have the receptor in the membrane that cytokines can bind to. That means that elicits two different signal transduction pathways. And here you get a response that's going to trigger them to kill infected cells. Here you get a response that's going to trigger these guys to divide and make plasma cells that will eventually secrete antibodies. Why is it that these two cells are both being communicated by the same signal molecule, the cytokine, but yet we have two different responses? Roman?
Because they have like the same receptor molecule. But like the proteins, their signal transduction pathways might be
That's correct.
different response.
Absolutely. So the receptors may be the same, therefore they can bind to the same signal molecule, which is the cytokine, but he said that the proteins in their signal transduction pathway may be different. So the ones activated here are different, which then will give you a different response. And absolutely, that's what's going on there. So that kind of pulls in chapter 11 here. And so that's the role of the helper T cells. The helper T cells are not in any way fighting them directly, anything to do with your infection. What they're doing is they are given the message that there is an infection by the APC, and then stimulate your two types of cells that will directly affect your antigens. By secreting antibodies or killing off the infected cells. So going back to this picture here, you don't have this next, I just put it in here to show. When we look at this, here's your helper T cell. So this is what we just talked about. Antigens engulfed and displayed by dendritic cells. That was this part right here. It's the antigens. They're engulfed and displayed by the APC, dendritic cell. That's going to activate the helper T cell. So that's what we have here. Then what does the helper T cell do? These red arrows, mine are color-coded, yours are not on your sheet, but these three red arrows represent the cytokines. So the helper T cell releases cytokines, and those cytokines stimulate three types of cells. They stimulate the cytotoxic T cells to eventually launch an attack against the infected cell. They stimulate themselves to make more helper T cells and therefore make more cytokines. Notice that they also make memory helper T cells. And so, so later on for your second infection, you already have some memory helper T cells. And then they stimulate your B cells. So that's what that refers to. And so these three arrows right here on this diagram are these three arrows here. So going to the helper T cell, cytotoxic T cell, and B cell. And so that's the role of the helper T cell. So then we have to look at this part here. So what happens to these cytokines that activate the cytotoxic T cell, and how do these guys eventually kill off your infected cells? All right, so that's this part here. So cytotoxic T cells, a response to infected cells. Cytotoxic T cells make what's called CD8, a surface protein that greatly enhances interaction between a target cell and a cytotoxic T cell. Binding to a class 1 MHC complex on an infected cell activates a cytotoxic T cell and makes it an active killer. The activated cytotoxic T cell secretes proteins that destroy the infected target cell. So that's what we have here in this picture. And so I did the same thing. I put a few captions underneath there to describe what's going on in this picture. So I'll give you a few minutes to look at that or write that down if you'd like, and then I'll talk about it. at this, your cytotoxic T cell is binding to, it says target cell. Your target cell is really your infected cell. Because remember, the cytotoxic T cell is going to kill any infected cells. Can somebody tell me, how is it that the cytotoxic T cell knows that this cell is infected? How does it know that it's infected, that target cell? That's your infected cell. Max, do you have an idea? On the outside of what? Well, so where's the pathogen here? This is the infected cell. So this is not the pathogen.
Oh, like the, I forget what they're called, the things on the inside.
The things on the inside. What?
Yeah, I don't know what they're called.
Okay. Well, what things are we talking about? So, like, what do you mean?
Oh, the antigen, the peptide antigens.
Okay, these guys here. That's right. These peptide antigens here are in the cell, and these guys were from the pathogen. So when a pathogen infects a cell, you have inside this affected cell the antigens that were from the pathogen. And remember, what an infected cell does is display that antigen on this MHC molecule. So the MHC molecule binds to it and displays this. So this is the antigen displayed. And the antigen is from the pathogen. And so this cell now has this foreign antigen displayed on it. A normal target cell, a normal cell won't have that. So this is the normal cell. There's no MHC displaying this abnormal antigen here. So remember, the antigen on the pathogen is different than any antigen on the surface of your cells. And so this is what targets this as a cell that's different. And so this is not a normal cell. And so that's a good thing. And so the cytotoxic T cell binds to it, to that complex, the MHC and the antigen presenting. This CD8 molecule just stabilizes it, makes it kind of stuck there to the target cell. And then, as we see here, the cytotoxic T cell, what this binding does is trigger the cytotoxic T cell to release this molecule called perforin. So it's already there. It releases that perforin, and it goes to the target cell, which forms holes in the target cell membrane, which then eventually triggers apoptosis and the cell to die. And so this cell here dies, and the cytotoxic T cell is released unharmed, and then that cytotoxic T cell can then be triggered by binding to go find another infected cell and bind to it and kill off it, and so on and so forth. So therefore, through this process, this is how the cytotoxic T cells attack and kill off the infected cells. So this is going on at the same time that your B cells are releasing antibodies to attack your pathogen, and any cells that have already been attacked by the pathogen are killed off by these cytotoxic T cells. So that's this part here. So the cytokines from the helper T cell, as well as the antigens displayed on the infected cells, stimulate your cytotoxic T cells. They divide and differentiate into memory and the active cells, and it's the active ones that are going to release that perforin and kill off infected cells, but also cancer cells and transplanted tissues. They basically can kill any cell that has different antigens than what you have on your cells. All right, and so transplanted tissues could be the case if an abnormal tissue that doesn't belong to you, if that has antigens on it that are not on your cells, your body will attack it just as if it was a pathogen. All right. All right, and then the last thing that we need to look at in terms of acquired immunity is, we go a little bit further with B cells here. So activation of B cells is aided by those cytokines and antigens binding to the helper T cells. Oh, there's that one.
Antibodies.
Yes, cross that out. I meant to cross that out. You don't need to know all these there's all these different classes of antibodies. It's beyond what you need to know for this class. Aided by cytokines and antigen binding to helper T cells, and then you go through clonal selection of B cells, generates the plasma cells that secrete antibodies. This is a review. I'm just going to give you one more little piece of information with the B cells. All right, I'm going to write some things down on this picture here. I didn't type anything out here. So this part here is a review of what we just looked at in a previous picture, that how your helper T cells are stimulated. So you have your macrophage that takes in the bacteria. This is the antigen. This is an antigen presenting cell, APC. Your macrophage takes in the bacterium. It's doing a good thing for you, destroying the bacterium. In the process, it displays that antigen. So this is the antigen displayed. And then a couple of things that are going to trigger the activation of a helper T cell. So notice here, from this picture to this picture, our helper T cell becomes activated. So, so we're going to write here what causes those to become activated. So in step number 1, so this says number 1 right here. So in step number 1 here, we have the APC, which is in this case is your macrophage, presents the antigen fragment. So why is that helpful? Because that antigen fragment is going to help activate this helper T cell. So the next thing that happens underneath here is the helper T cell is activated, but it's activated by a couple of different things. It's activated by actually binding to the antigen displayed by the APC. So it's activated by binding, I should say activated by, the bullet here, binding to the antigen, I'll put MHC complex. And then the second thing that activates it is something that they didn't illustrate here that we talked about in the previous picture. When these guys bind, the APC releases something. What does it release? It releases those cytokines here. So it releases, I'm going to draw that in here. And the cytokines activate, also help to activate the helper T cell. So I'm going to put the second thing is cytokines from the APC. So that's what's going on in that picture here. So then when we look at, as a result of that, now we have an activated helper T cell. Now notice here, the activated helper T cell is binding to a B cell. Now what we've talked about already with B cells, we're going to run out of time, aren't we? With B cells is that B cells can divide into plasma cells that secrete antibodies and memory cells. We're going to see here, we're adding a new piece of information here, that B cells can actually uptake the pathogen themselves and display part of the antigen. All right, so that's a new piece of information here. I don't know if I can write that down here real quick before the end of the hour, but let's do this real quick. 2. All right, 2. B cells take in pathogen and display its antigen. What happens next is that the helper T cell binds to the B cell. And releases cytokines. So the helper T cell binds to the B cell, releases cytokines, both of which activate the B cell. Activating B cell. So that's this part right here is the new part to this. And that and the activation of the B cell causes the dividing and differentiating, which we've already talked about.